Hello everyone, ಹೇಗಿದ್ದೀರಾ ಮಕ್ಕಳೇ? I hope you all are keeping great. Welcome to PW Kannada and welcome to NCERT line by line series. So ಇವತ್ತು ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡುವಂತಹ ಚಾಪ್ಟರ್ ಬಯೋಟೆಕ್ನಾಲಜಿ and its application. ನಾನು ನಿಮ್ಮ ವಿಂಧ್ಯಾ ರಾವ್ ಮ್ಯಾಮ್. ಇವತ್ತು ನಾನು ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ಯಾವ ಲೈನ್ ಇಂಪಾರ್ಟೆಂಟ್, ಯಾವ ಲೈನ್ ಇಂದ ಕ್ವೆಶ್ಚನ್ಸ್ NEET 2023 ಅಲ್ಲಿ ಇರಲಿ ಅಥವಾ ಅದಕ್ಕೂ ಮುಂಚೆ ಕ್ವೆಶ್ಚನ್ಸ್ ಬಂದಿದೆ and which concept you have to for sure study and go ಅಂತ ಇವತ್ತು ನಾನು ಹೇಳಲಿಕ್ಕಿದ್ದೇನೆ. ಸೊ ಲೆಟ್ಸ್ ಗೆಟ್ ಸ್ಟಾರ್ಟೆಡ್ ವಿದೌಟ್ ವೇಸ್ಟಿಂಗ್ ಮಚ್ ಟೈಮ್. ಸೊ ಇನ್ನೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ವಿಚಾರ ಹೇಳಲೇಬೇಕು. ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡಿದ್ದಾರೆ ಅಂದ್ರೆ ಒಂದು ಕಾನ್ಸೆಪ್ಟ್ ಅನ್ನ ಎಡಿಷನಲ್ ಆಗಿ ಆಡ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ಇದು ನಿಮ್ಮ ಹಳೆ NCERT ಅಲ್ಲಿ ಇರ್ಲಿಕ್ಕಿಲ್ಲ, ಆದ್ರೆ ಹೊಸ NCERT ಅಲ್ಲಿ ದೇ ಹ್ಯಾವ್ ಆಡೆಡ್ ಅ ಕಾನ್ಸೆಪ್ಟ್. ಸೊ ವಿ ವಿಲ್ ಆಲ್ಸೋ ಟಾಕ್ ಅಬೌಟ್ ದಟ್ ಕಾನ್ಸೆಪ್ಟ್. ಸೊ if you see the introduction part of this chapter, we already know the application of biotechnology. So adrane helidare illi nodi, biotechnology as you would have learned from the previous chapter essentially deals with the industrial scale production of biopharmaceuticals and biological using GM, that is genetically modified microbes, fungi, plants and animals. So namige idu gottiruvantaha vicharave, that is biotechnology application ಏನು main agi andre that is to make the certain biopharmaceuticals and using the GM, that genetically modified organisms like fungi, microbes, plants and animals. Okay, so this we already know. So adike nanu punaha enu separate agi underline madlikkilla. I'm not going to do any underlining for the application. Now another additional thing that they have added in this particular chapter yenandre, so they have told what is the different applications of biotechnology. So biotechnology na ನಾವು ಯಾವುದೆಲ್ಲ ಬೇರೆ ಬೇರೆ ಏನು ಇದಕ್ಕೆ ಯೂಸ್ ಮಾಡಬಹುದು ಅಂತ ಹೇಳಿದ್ದಾರೆ. ಸೊ ಅದನ್ನ ನಾನು ಇಲ್ಲಿ ಅಂಡರ್ಲೈನ್ ಮಾಡ್ಲಿಕ್ಕಿದ್ದೇನೆ ಬಿಕಾಸ್ ಇದರಿಂದ ಕ್ವೆಶ್ಚನ್ ಬರೋ ಚಾನ್ಸಸ್ ಇರುತ್ತೆ, ಆದ್ರೆ ಇಷ್ಟವರೆಗೆ ಕ್ವೆಶ್ಚನ್ ಬರಲಿಲ್ಲ. ಸೊ ಅಪ್ಲಿಕೇಶನ್ ಆಫ್ ಬಯೋಟೆಕ್ನಾಲಜಿ ಈಸ್ ಇನ್ ಮೇಕಿಂಗ್ ಆಫ್ ಥೆರಪೆಟಿಕ್ಸ್, ದಟ್ ಇಸ್ ಫಾರ್ ದ ಮೆಡಿಸಿನಲ್ ಪರ್ಪಸ್ ಟು ಮೇಕ್ ಸರ್ಟನ್ ಮೆಡಿಸಿನ್ಸ್. ಡಯಾಗ್ನಾಸ್ಟಿಕ್ಸ್ ಅಂದ್ರೆ the few things which is being used to diagnose, that is to detect certain diseases, adike diagnostics anta heluvantaddu. Genetically modified crops for agriculture purpose. In the processed food we use biotechnology. For bioremediation, bioremediation ಗೆ example ಏನು ಮ್ಯಾಮ್ ಅಂತ ಕೇಳಿದ್ರೆ in the case of oil spillage, oil ಅನ್ನ ಕನ್ಸ್ಯೂಮ್ ಮಾಡುವಂತಹ ಬ್ಯಾಕ್ಟೀರಿಯಾವನ್ನ ಸೈಂಟಿಸ್ಟ್ ಕಂಡು ಹುಡುಕ್ತಾರೆ. that is they will be genetically modifying Pseudomonas putida which can eat up, which can consume oil spillage. So adu ಒಂದು example for bioremediation. And in the case of waste treatment and energy production. So idu bere bere applications of biotechnology. Okay, and coming to the next part, that is the critical research area of biotechnology. idu NEET ಎಕ್ಸಾಮಿಗೆ ಎಷ್ಟವರೆಗೆ ಬರಲಿಲ್ಲ, ಆದ್ರೆ ನಿಮ್ಮ ಸೆಕೆಂಡ್ PUC ಅಲ್ಲಿ ಏನ್ ಮಾಡಿದ್ರು, ಮಾಡೆಲ್ ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್ ಅಲ್ಲಿ ಕ್ವೆಶ್ಚನ್ ಕೇಳಿದ್ರು 2 ಮಾರ್ಕರ್ 3 ಮಾರ್ಕರ್ ಆಗಿ ಏನು, ದಟ್ ಇಸ್ ವಿಚ್ ಆರ್ ದ 3 ಕ್ರಿಟಿಕಲ್ ರಿಸರ್ಚ್ ಏರಿಯಾಸ್ ಆಫ್ ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಂತ. ಸೊ ದಟ್ ಇಸ್ these 3, that is providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme, that is to make the catalyst or an enzyme, creating optimal condition to make such catalyst, biocatalyst, in order to act. And the third is downstreaming processing. So these are the 3 critical research areas of biotechnology. ಇದನ್ನ ನಾನು ಪುನಃ ಅಂಡರ್ಲೈನ್ ಮಾಡೋದಿಲ್ಲ ಬಿಕಾಸ್ ಇಟ್ಸ್ ನಾಟ್ ರಿಯಲಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ದ NEET ಎಕ್ಸಾಮ್. ಅಂಡ್ ದೆನ್ right now we will be studying about biotechnological application in agriculture. Agriculture ಅಲ್ಲಿ ಹೇಗೆ biotechnology is helpful ಅಂತ. ಸೊ ಅದಕ್ಕೂ ಮುಂಚೆ why are we using biotechnology in agriculture? That is to increase the food production. Okay, so let us take a look at the 3 options that can be thought to increase the food production. So ಯಾವುದೆಲ್ಲ ಬೇರೆ ಬೇರೆ ಟೆಕ್ನಿಕ್ಸ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಫುಡ್ ಪ್ರೊಡಕ್ಷನ್ ಅನ್ನ ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಟ್ರೈ ಮಾಡಿದ್ದಾರೆ ಸೈಂಟಿಸ್ಟ್ ಅಂತ ಕೇಳಿದ್ರೆ, that is going to be one is agrochemical based agriculture. ಅಂದ್ರೆ ಕೆಮಿಕಲ್ ಫರ್ಟಿಲೈಸರ್, ಪೆಸ್ಟಿಸೈಡ್ಸ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು they have tried to increase the food production. And the next is with the help of organic agriculture and genetically engineered crop based agriculture. So these are the 3 ways to increase the food production. Okay, the most important here is yaudu, that is genetically engineered crop based agriculture, and other 2 are organic culture and using chemical fertilizers. So green revolution anna use madkondu scientists tried to increase the food production triple. So the green revolution succeeded in tripling the food supply, but it was not enough for as per the demand. Okay, so however they also tried increasing the food production using fertilizers and pesticides. So it's already discussed ಮಾಡಿ ಆಗಿದೆ. Okay, and as traditional breeding techniques fail to keep the pace with the demand to provide sufficiently fast and efficient system for crop improvement, another technology called as tissue culture got developed to increase food production. So organic farming use ಮಾಡಿಕೊಂಡು chemical fertilizers ಅಂದ್ರೆ agrochemical based agriculture use ಮಾಡಿಕೊಂಡು they tried increasing the food production, and yes, of course, now we have genetically modified crops or genetically engineered crops to increase the food production. But scientists also tried to increase food production with the help of tissue culture. So tissue culture andre ಏನು, that is whole plant could be regenerated from the ex-plant, that is any part of the plant can be taken out and grown in a test tube under a sterile condition in a special nutrient media is called as tissue culture. So tissue culture andre actually ಏನು, so entire whole plant can be produced with the help of ex-plant. Ex-plant andre ಏನು, it can be the any part of the plant like a root, stem, leaf or anything like that. Okay, so whole plant could be regenerated from the ex-plant, that is any part of the plant taken out and grown in a test tube under a sterile condition in a nutrient rich media is called as tissue culture. So idu additional add ಆಗಿರುವಂತಹ concept for your new NCERT, ಇದು ಹೊಸ NCERT ಅಲ್ಲಿ ಇರುವಂತಹ ಕಾನ್ಸೆಪ್ಟ್. ಓಕೆ, ಸೊ ಯಾಕೆ ನಾವು ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಅನ್ನ ನೋಡಬಹುದು ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಅಂದ್ರೆ ಬಿಕಾಸ್ ಆಫ್ ದ ಸ್ಪೆಷಲ್ ಕೆಪಾಸಿಟಿ ಆಫ್ ದ plant, that is totipotency. So this capacity to generate a whole plant from any cell or explant is called as totipotency. Totipotency ನಾವು ಬರೀ plants ಅಲ್ಲಿ ಮಾತ್ರ ನೋಡುವಂತಹ property, not in any animals. You will learn how to accomplish this in higher classes. So detail ಆಗಿ ನೀವು higher class ಅಲ್ಲಿ ನೋಡ್ಲಿಕ್ಕಿದ್ದೀರಿ. It is important to stress here that the nutrient medium must provide a carbon source such as sucrose, also inorganic salts, vitamins, amino acids and growth regulators like auxin, cytokines. So idistu nutrient media dalli irbeku if you are performing tissue culture of certain explant or any plant. Okay, sugar source yavdu, sucrose irbeku, and rest should be there in the growth regulators to increase the growth, cytokines, auxins alla. By application of these methods, it is possible to achieve propagation of large number of plants in a very short duration, and this method of producing thousands of plants through the tissue culture is called as micropropagation. Micropropagation andre enu, as the name say, micro, with the help of very small part of the plant we are able to propagate, we are able to increase the number of plants to thousands of plants. So application of this method is possible to propagate a large number of plants in a very short duration of time, and ಇದಕ್ಕೆ ನಾವು ಹೇಳುವಂತದ್ದು micro propagation ಅಂತ. So this is the definition of micro propagation, that is a method of producing thousands of plants with the tissue culture. So each of these plants will be genetically identical to the original plant from which they were grown, that is they are called as soma clones. So ಇದರ tissue culture ಮುಖಾಂತರ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರುವಂತಹ plants ಏನಿದೆ, they are identical to the original parental plant. ಅದಕ್ಕೆ ನಾವು ಇಂತಹ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ, ಇಂತಹ ಚಿಕ್ಕ ಚಿಕ್ಕ ಪ್ಲಾಂಟ್ಸ್ ಏನು ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರುತ್ತೆ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಮುಖಾಂತರ, ಇದಕ್ಕೆ ನಾವು ಸೋಮಾ ಕ್ಲೋನ್ಸ್ ಅಂತ ಕರಿಬಹುದು. ಯಾಕೆ ಸೋಮಾ ಕ್ಲೋನ್ಸ್ ಅಂತ ಹೆಸರು? ಕ್ಲೋನ್ಸ್ ಅಂದ್ರೆ ಏನು? ಐಡೆಂಟಿಕಲ್. ಸೋಮಾ, ದಟ್ ಇಸ್ ಸೊಮ್ಯಾಟಿಕ್ ಸೆಲ್ಸ್ ಏನಿದೆ, ಇಟ್ ಇಸ್ ಐಡೆಂಟಿಕಲ್ ಟು ದ ಸೊಮ್ಯಾಟಿಕ್ ಸೆಲ್ಸ್ ಆಫ್ ದ ಪೇರೆಂಟಲ್ ಪ್ಲಾಂಟ್. ಸೊ ಅದಕ್ಕೆ ಇಂತಹ ಐಡೆಂಟಿಕಲ್ ಏನು ಪ್ಲಾಂಟ್ಸ್ ಏನು ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ, ಇದಕ್ಕೆ ನಾವು ಸೋಮಾ ಕ್ಲೋನ್ಸ್ ಅಂತ ಕರಿಬಹುದು. Okay, so this is the definition of soma clones. Soma clones andre enu anta. So many important food plants like tomato, banana, apple. So idella tissue culture mukantara commercialize ಮಾಡಿ we are able to produce in laboratory through tissue culture. Okay, so this is what you have to know about tissue culture from this chapter. So ಏನೆಲ್ಲ ಬೇಕು ಅದನ್ನ ಮಾತ್ರ ನಾನು ಅಂಡರ್ಲೈನ್ ಮಾಡಿದ್ದೇನೆ. Okay, nothing more, nothing less. What is important I have already underlined. And then we have got something more that is about tissue culture only. Another important application of the method, that is tissue culture na innondu application ಏನಂದ್ರೆ recovery of healthy plants from the diseased plant. So let's say that we have got a plant which is deceased. Adre intaha plant inda we want to produce the disease-free plant. Okay, let's say we have a tomato plant, a very good variety of tomato plant, and it is deceased. Adre intaha tomato plant in the we want the another tomato plant, and we should be able to produce a lot of tomatoes. So in this case, we have a like there is an application of the tissue culture to produce such disease-free plants from the deceased plants. So another important application of the method is the recovery of the healthy plants from the diseased plants. Even if the plant is infected with a virus nantaha deadly pathogen effect adru saha we are able to recover, that is through the meristem, because the meristem of the diseased plant is going to be free of the pathogen, free of the virus. Hence one can remove the meristem and grow it in vitro, that is they can grow in the laboratory to obtain virus-free plants, and scientists have succeeded in culturing meristems of banana, sugarcane, potato. So yaudella plants anna yaudella diseased, yaudella varieties of plants, even when they are deceased, from which we can take the meristem and further grow, that is potato, banana and sugarcane. Okay, so examples for tissue culture, yaudella plants alli tissue culture madidare, yaudella plants alli meristem culture madidare. So idike ನಾವು ಏನಂತ ಕರೆಯುವಂತದ್ದು meristem culture. Okay, because the meristem of the infected plants are disease free. So meristem culture yaudella plants alli madidare, that is sugarcane, banana, potato. Ad hege maduvanತದ್ದು ಏನು application, so we have already seen. So scientists have even isolated single cells from the plants, and after digesting their cell walls have been able to isolate naked protoplast. Isolated protoplast from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplast, which can be further grown to form a new plant, and these hybrids are called as somatic hybrids. So somatic hybridization or somatic hybrids ಏನು ಅಂತ ಈ ಪ್ಯಾರಾಗ್ರಾಫ್ ಅಲ್ಲಿ ಇದೆ. So that is about somatic hybrid production. ಏನು actually somatic hybrid ಅಂದ್ರೆ, so protoplast of a particular ಏನು plant ಅನ್ನ ನಾವು ತಗೊಳ್ಳುವಂತದ್ದು, that is by digesting the plant part, so that the cell wall, when we digest, the rest of the living part of the cell is called as protoplast. ಸೊ ಅಂತಹ ಒಂದು ಪ್ಲಾಂಟಿನ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಅನ್ನ ಇನ್ನೊಂದು ತರದ ಪ್ಲಾಂಟಿನ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಜೊತೆಗೆ ಫ್ಯೂಸ್ ಮಾಡುವಾಗ ವಿ ಆರ್ ಏಬಲ್ ಟು ಗೆಟ್ ದ ಟೂ ವೆರೈಟೀಸ್ ಗೆಟಿಂಗ್ ಮಿಕ್ಸ್ಡ್, ಅಂಡ್ ಅಂತಹ ಹೊಸ ಏನು ಫ್ಯೂಸ್ಡ್ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಏನಿರುತ್ತೆ, ಸೊ ಆ ಪ್ಲಾಂಟ್ ಏನು ಫರ್ದರ್ ಡೆವಲಪ್ ಆಗುತ್ತೆ, ದೇ ವಿಲ್ ಬಿ ಶೋಯಿಂಗ್ ದ ಏನು ಫೀಚರ್ಸ್ ಆಫ್ ಬೋತ್ ಆಫ್ ದೀಸ್ ಟೂ ವೆರೈಟೀಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ಸ್. ಸೊ ಇಂತಹ ಇದು ಹೈಬ್ರಿಡ್ ಅಂತ ಕರಿಬಹುದಲ್ವಾ, ಇಂತಹ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಫ್ಯೂಷನ್ ಗೆ ನಾವು ಹೈಬ್ರಿಡ್ ಅಂತ ಕರಿಬಹುದು, ಯಾಕಂದ್ರೆ the protoplast would be having the two different varieties of features. So then we can call such resultant plant or protoplast as a hybrid. So ಇದಕ್ಕೆ ನಾವು somatic hybrid ಅಂತ ಹೇಳುವಂತದ್ದು, and the process is called as somatic hybridization. Okay, ಯಾಕೆ ಹೆಸರು somatic? Because the protoplast will be having the somatic feature, like they are a somatic cell alva. So we know that in any type of a plant or human being or an animal, we are able to see two types of cells, somatic ಮತ್ತೆ sex cells. Somatic, soma ಅಂದ್ರೆ ಏನು, the body cells. Sex cells ಅಂದ್ರೆ ಏನು, the one that is present in the reproductive organs. So protoplast is actually the somatic cells. So intaha hybrid ಏನು produce ಆಗುತ್ತೆ through these two protoplasts, they are called as somatic hybrid, and the process is called as somatic hybridization. Simple. So imagine a situation when a protoplast of tomato is fused with that of the potato, then we can call such new hybrid as pomato alva. So well, this has been achieved, resulting in the formation of pomato. Unfortunately, this plant did not have all the desired combination of characteristics for its commercial utilization. Commercially we couldn't commercial ಆಗಿ ಇದನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಮಾರ್ಕೆಟ್ ಮಾಡುವಂತಹ ಗ್ರೇಟ್ ಫೀಚರ್ಸ್ ಈ ಪೊಮ್ಯಾಟೋಗೆ ಇರಲಿಲ್ಲ. ಸೊ ದಟ್ ಇಸ್ ವೈ ಇಟ್ ವಾಸ್ ನಾಟ್ ಏಬಲ್ ಟು ಗೆಟ್ that much of attention. ಸೊ ಪೊಮ್ಯಾಟೋವನ್ನ ಹೇಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರು, that is through somatic hybridization, because they have taken the protoplast of the tomato plant and protoplast of the pomato plant, tomato and potato. Okay, so that is about somatic hybridization. So this was the additional concept that was being added in the new NCERT. ಇದು ಹಳೆ NCERT ಅಲ್ಲಿ ಇಲ್ಲ. Okay, now next Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield from their fields? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment can be reduced? Use of genetically modified crops is a possible solution for all of these things. So, adra ಬಗ್ಗೆ ನಾವಿವತ್ತು ಕಲಿಲಿಕ್ಕಿದ್ದೀವಿ, ನೋಡ್ಲಿಕ್ಕಿದ್ದೀವಿ. So, plants, bacteria, fungi, animals, whose genes have been altered, manipulated, are called a genetically modified organisms. So, ಯಾವುದೆಲ್ಲ genetically modified organisms under ಬರುತ್ತೆ? That is plants, bacteria, fungi, animals. Already we have seen in the previous slide as well. Alva? So, idishtu genetically modified organisms anna madidare by altering the genes of plant, animals, fungi and bacteria. Okay, so what are the different uses of this genetically modified plants specifically? So, adranna they have given in the form of list. Idralli bari ಒಂದು ಅಥವಾ ಎರಡು ಪಾಯಿಂಟ್ ತುಂಬಾನೇ ಇಂಪಾರ್ಟೆಂಟ್. Rest all are okay points because questions ಬರಲಿಲ್ಲ. ಆದರೂ ಸಹ you can expect question from these points. So, ಇದು important. ಯಾಕೆ ಇದು important? ಒಂದು ಸಲ ಒಂದು question ಬಂದಿದೆ related to golden rice. Okay. However, we will be discussing about each of this point in detail in this chapter, but still have a look on this. So, ಯಾವುದೆಲ್ಲ different importance of genetically modified plants ಅಂತ ಈ paragraph ಅಲ್ಲಿ ಇದೆ with the help of 5 points. So, the very important thing, why are we making genetically modified plants? That is, the crop has been made more tolerant to abiotic stress. So, with the help of genetically modified plants, we are able to make certain crops more tolerant for the abiotic stress. Yaudella abiotic stress? Drought, extreme water or extreme salt, heat. So, idella abiotic stress agirutte. Cold, drought, salt, heat. So, genetically modified plants are being made to overcome or tolerate abiotic stresses. Important. And the next use is reduced reliance on chemical pesticides. So, we have made certain genetically modified crops or plant which is the pest resistant. Okay. So, ee tara navu we can prevent the use of chemical fertilizers, help to reduce post harvest loss. So, idella point KCT exam ಗೆ ಬಂದಿದೆ ತುಂಬಾ ಸಲ, ಆದರೆ NEET ಅಲ್ಲಿ ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಆದರೂ ಕಲ್ತು ಹೋಗಿ, ಬೆಟರ್ ಟು ಲರ್ನ್ ಆಲ್ ದಿಸ್ ಥಿಂಗ್ಸ್. Increased efficiency to mineral usage by the plants through GM plants. We have made increased efficiency of mineral usage. So, it will be preventing the early exhaustion of the fertility of the soil. And the last and most important, NEET 2019 ಅಲ್ಲಿ ರಿಪೀಟ್ ಆಗಿರುವಂತಹ ಕ್ವೆಶ್ಚನ್ ನೋಡಿ, that is enhanced nutritional value of the food is the last but most important use of genetically modified plants. Okay, so golden rice is an example which is rich with vitamin A that is helping in preventing night blindness. So, GM plants are being used to enhance the nutritional value of the food. Okay, now, so let's get started with each of these importance one by one. So, one by one agi navu importance anna nodta hogwa of all of these different genetically modified plants. Okay, in addition to these uses, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, pharmaceuticals. Hada NCERT alli illa anta ankondiddene. In addition to these, genetically modified crops are being used to make certain alternative resources to industries. That is, just starch anna produce ಮಾಡುವಂತ genetically modified plants anna produce ಮಾಡಿದ್ದಾರೆ. ಜಾಸ್ತಿ fuels produce ಮಾಡ್ಲಿಕ್ಕೆ help ಆಗಿ, like they have made certain genetically modified plants. Pharmaceuticals ige. So, these are the other additional use of genetically modified crops and plants. So, some of the application of biotechnology in agriculture that you will study in the detail is given over here. That is about BT cotton, which is very important. NEET 2020 ಎರಡು ಸಲ ಬಂದಿದೆ. NEET 2020 ಅಲ್ಲಿ, NEET 2019 ಅಲ್ಲಿ ಒಂದು ಸಲ ಬಂದಿದೆ. ಕಂಬೈನ್ಡ್ ಕ್ವೆಶ್ಚನ್ ಆಗಿ ಸಹ NEET 2021 ಅಲ್ಲೇನೋ ಕ್ವೆಶ್ಚನ್ ಕೇಳಿದ್ರು ಫ್ರಮ್ BT cotton. BT ಅಂದ್ರೇನು? Bacillus thuringiensis. So, BT toxin is produced by the bacteria Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria and been expressed in the plants to provide resistance to insect without the need for any insecticides. In effect, created biopesticides. So, ee tara biopesticides are being made with the help of genetically modified plants by this gene, Bt gene, that is present naturally in the Bacillus thuringiensis bacteria. So, aa gene anna nama plant which is affected by insect. So, a plant ge haktare through Agrobacterium tumefaciens plasmid. Okay. Bt cotton. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as Lepidopterans, tobacco budworm, armyworm, coleopterans, beetles, diphterans, flies, mosquitoes. So, this all can be killed by varieties of the Bt genes through their Bt toxins. Bacillus thuringiensis forms protein crystal during a particular phase of their growth. These crystals will be containing the toxic insecticidal protein. Adre ee toxin, it won't be killing the Bacillus bacteria. Why? Yakandre this Bacillus thuringiensis Bt toxin protein will be existing in inactive protoxin form. But once an insect ingests this inactive toxin, that is an active protoxin, it is then converted immediately into the active form because of the alkaline pH of the gut, which will be solubilizing the crystals. That is, it will be solubilizing the Bt protoxin to Bt toxin. Pro andrenu inactive toxin, protoxin andre active toxin. So, the activated toxin will be binding then to the surface of the midgut epithelial cells, and it will be then creating the pores that will be causing the cell to swell and lysis and eventually cause death of such insect. So, that is about Bt toxin and Bt cotton. Yake Bt cotton? We will see. So, ಈ ವಿಡಿಯೋದಲ್ಲಿ ಸ್ವಲ್ಪ ವಾಯ್ಸ್ ಇಶ್ಯೂ ಇರಬಹುದು. Please don't mind about it. Okay. So, Bt cotton anta ನಾವು ಏನು ಕಲಿತೀವಿ. So, we are actually inserting this particular gene which is able to produce Bt protoxin or Bt toxin in specifically cotton. So, specific Bt toxin gene were isolated from the Bacillus thuringiensis and incorporated into several crops such as cotton. The choice of genes depends upon the crop and the targeted pest, as most Bt toxin are insect group specific. The toxin is coded by the gene 𝐬1AC named 𝐬. There are a number of them. For example, the protein encoded by the genes 𝐬1AC, 𝐬2AB will be controlling cotton bollworm, 𝐬1AB will be controlling corn borer, and idu tumbane important. So, NEET 2020 alli 2 sala question bandide. NEET 2019 alli 1 sala question bandide. NEET 2020 alli combined question ಆಗಿಯೂ 1 sala question bandide related to Bt toxin. So, Bt toxin anta yake hesaru? Because it is Bacillus thuringiensis bacteria the toxin. This is also called as 𝐬 protein because they will be present in the form of a crystal protein, that is inactive protoxin form alli irutte. Adikke idrana 𝐬 protein anta karitare, Bt protoxin anta karitare. They will be activated in the alkaline pH of the insect gut. Okay. ಯಾವ ಜೀನ್ ಯಾವ ಪ್ಯಾಥೋಜನಿಗೆ ಸ್ಪೆಸಿಫಿಕ್ ಅಂತ ಗೊತ್ತಿರಬೇಕು. 𝐬1AC, 𝐬2AB is for cotton bollworm. 𝐬1AB controls corn borer. Okay. And about like yava bere bere insect, bere bere arthropods they can kill. So, that also you should be knowing. Ashte. And yava plant alli they have incorporated and tried this, that is Bt cotton, Bt corn, rice, tomato, potato and soya bean. So, idu saha important things to note. Okay. And then we have got another pest resistant plant through the mechanism RNAi athava interference RNA or RNA interference or RNA silencing or double stranded RNA. So they have got many names. NEET 2020 ಅಲ್ಲಿ 2 ಸಲ ಕ್ವೆಶ್ಚನ್ ಬಂದಿದೆ. NEET 2019 ಅಲ್ಲಿ ಪುನಃ 2 ಸಲ ಕ್ವೆಶ್ಚನ್ ಬಂದಿದೆ from this concept RNA interference. This is very important concept for the NEET exam from this chapter. Several nematodes, that is parasites. Before this we saw the arthropod, right? The phylum arthropod, the class insect. But now the pest here is nematode, a parasite, a wide variety of plants and animals. Several nematodes, parasites in plants and animals, including human beings. A nematode called as 𝐊𝐀𝐍𝐍𝐀𝐃 will be infecting the roots of the tobacco plants, and it will be causes the great reduction in the yield of the crops. So a novel strategy was adopted to prevent this infestation, which was based upon the process of RNA interference or RNAi, because it will be interfering with the conversion of RNA to protein. It will be interfering with the translation mechanism. Aita? RNAi will be taking place in all the eukaryotic organisms. Yake? As a method of cellular defense. So, ee method enide idu ella eukaryotic cells alli aguvantadu as a cellular defense mechanism, and this method will be involving silencing a specific mRNA due to a complementary double stranded RNA. So ನಿಮಗೆ ಗೊತ್ತು RNA ಯಾವತ್ತು ಸಿಂಗಲ್ ಆಗಿರಬೇಕು ಅಂತ. ಸೊ ಎಲ್ಲಾದ್ರೂ RNA ಡಬಲ್ ಆಯ್ತು ಅಂದ್ರೆ ಇಟ್ ಮೀನ್ಸ್ there is no further translation that will be happening, protein production ಆಗುದಿಲ್ಲ ಅಂತ ಅರ್ಥ. ಸೊ ಇಲ್ಲಿ RNAi mechanism will be happening because of the double stranded RNA formation, because of the complementarity between the two RNA. So ಎರಡು RNA which are complementary to each other. So ಇಂತಹ ಎರಡು RNA produce ಆಗೋದ್ರಿಂದ there is going to be silencing of such RNA so that no more protein production will be happening. Okay, the source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements, that is transposons that replicate via an RNA intermediate. So NEET 2022 ಅಲ್ಲಿ question ಕೇಳಿದ್ದಾರೆ related to transposons. Very important. So ಯಾಕೆ ನ್ಯಾಚುರಲ್ ಆಗಿ ಎಲ್ಲ ಯುಕ್ಯಾರಿಯೋಟಿಕ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ RNAi mechanism ಆಗುವಂತದ್ದು? that is to prevent the virus RNA production, to prevent the transposons. Transposons ಇಂದ ಏನಾಗುತ್ತೆ? ಮೊಬೈಲ್ ಜೆನೆಟಿಕ್ ಎಲಿಮೆಂಟ್ಸ್ ಇಂದ ಏನಾಗುತ್ತೆ? they are also called as jumping genes. So ಇದರಿಂದ ಮ್ಯೂಟೇಷನ್ ಆಗಬಹುದು, ಕ್ಯಾನ್ಸರ್ ಆಗಬಹುದು. So in order to prevent this, we have the natural cellular defense mechanism, that is RNAi. So source of this complementary RNA could be from an infection by the viruses having RNA genomes or the mobile genetic elements that will be replicating via RNA intermediate. So source of RNA interference is naturally of the cause of the virus and cause of the mobile genetic elements. Using agrobacterium vector, nematode specific genes were introduced into the host plant. So nematode specific DNA anna host ಒಳಗಡೆ ಹಾಕ್ತಾರೆ with the help of agrobacterium. The introduction of the DNA was such that it produced both sense and antisense RNA in the host cells. So ಎರಡು ತರದ RNA ನ produce ಮಾಡುತ್ತೆ so that they can show complementarity and they can silence. Okay. So these two RNAs being complementary to each other they will be forming the double stranded RNA. These two RNAs being complementary to each other will be forming double stranded RNA. That will be initiating RNAi mechanism and thus silenced. That's how they will be silencing the specific mRNA of the nematode as well. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. So ಇಲ್ಲಿ NCERT ಅಲ್ಲಿ ಇಮೇಜ್ ಅನ್ನು ಕೊಟ್ಟಿದ್ದಾರೆ. ಹೋಸ್ಟ್ ಪ್ಲಾಂಟ್ generated double stranded triggers protection against nematode infestation. This A image is roots of typical control plant, that is root of the plant which is having the nematode, that is which is having the root knots. This is transgenic plant which is having the nematode specific gene in them, which is having the RNAi mechanism against the nematode. So transgenic plant roots 5 days after deliberate infestation of nematode, but protected through the novel mechanism of RNAi. So ಇಲ್ಲಿ ನೀವು ಯಾವುದೇ ತರದ ರೂಟ್ ನಾಟ್ಸ್ ಅನ್ನ ನೀವು ಕಮ್ಮಿ ನೋಡ್ತೀರಾ. ಇಲ್ಲಿ ಜಾಸ್ತಿ ರೂಟ್ ನಾಟ್ಸ್ ಇದೆ, ಇಲ್ಲಿ ಕಮ್ಮಿ, ತುಂಬಾ ಕಮ್ಮಿ ಇದೆ. Because this is a transgenic plant which is having the novel mechanism. So ಇಲ್ಲಿ ಏನು how they are being made transgenic is that is by introducing the nematode specific DNA, so that the natural RNAi mechanism will go and specifically silence when the real nematode enters. Okay, so idu related to the nematode ಆಯ್ತು. ಇದಕ್ಕೂ ಮುಂಚೆ ನಾವು ನೋಡಿರುವಂತದ್ದು BT cotton. ಸೊ ಈ ಇಮೇಜ್ ಏನು ಹೇಳುತ್ತೆ, ಕಾಟನ್ ಬಾಲ್ ಅನ್ನು ತೋರಿಸ್ತಾ ಇದೆ. ಇದು A is destroyed by the ball worm and A is fully mature cotton worm. This is destroyed by the ball worm. So ಇಲ್ಲಿ ನೀವು ನೋಡಬಹುದು ಕಾಟನ್ ಬಾಲ್ಸ್ ಅಲ್ಲಿ there is a hole because of the ball worm, and this is mature, fully matured cotton balls without any infestation. Okay, so that is about pest resistant plant, BT cotton and RNAi mechanism. Okay, next is application of biotechnology in medicine. So idra under barovanthaddu making of genetically engineered insulin. So we'll see some fact. Recombinant DNA technology process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Furthermore, the recombinant therapeutics do not induce unwanted immunological responses as in common case of similar products isolated from the non-human sources. At present, about 30 recombinant therapeutics have been approved for human use all over the world. In India, 12 of these are presently being marketed. So this is all you should be knowing from the small introduction part. ನಂತರ ಬರುವಂತದ್ದು ಪ್ರಾಪರ್ ಅಪ್ಲಿಕೇಶನ್ ಅಂಡರ್ ಮೆಡಿಸಿನ್, ದಟ್ ಇಸ್ ಮೇಕಿಂಗ್ ಆಫ್ ಇನ್ಸುಲಿನ್, ಜೆನೆಟಿಕಲಿ ಇಂಜಿನಿಯರ್ಡ್ ಇನ್ಸುಲಿನ್. ನೀಟ್ 2020 ಅಲ್ಲಿ ಕ್ವೆಶನ್ ಕೇಳಿದ್ದಾರೆ, ನೀಟ್ 2021 ಅಲ್ಲಿ ಕ್ವೆಶನ್ ಕೇಳಿದ್ದಾರೆ ರಿಲೇಟೆಡ್ ಟು ದಿಸ್ ಕಾನ್ಸೆಪ್ಟ್. ಸೊ ಕ್ವಿಕ್ ಆಗಿ ನಾವು ಒಂದ್ಸಲ ನೋಡ್ತಾ ಹೋಗುವ. ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ಆಫ್ ಅಡಲ್ಟ್ ಆನ್ಸೆಟ್ ಡಯಾಬಿಟೀಸ್ ಇಸ್ ಪಾಸಿಬಲ್ ಬೈ ಟೇಕಿಂಗ್ ಇನ್ಸುಲಿನ್ ಎಟ್ ರೆಗ್ಯುಲರ್ ಟೈಮ್ ಇಂಟರ್ವಲ್ಸ್. ನಮಗೆ ಗೊತ್ತದು. ವೈ ವುಡ್ ಅ ಡಯಾಬಿಟಿಕ್ ಪೇಷಂಟ್ ಡೂ ಇಫ್ ಇನಫ್ ಹ್ಯೂಮನ್ ಇನ್ಸುಲಿನ್ ವಾಸ್ ನಾಟ್ ಅವೈಲೇಬಲ್? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. ಆದರೆ ಬೇರೆ ಆನಿಮಲ್ಸ್ ಇಂದ ನಾವು ಇನ್ಸುಲಿನ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡಿದ್ರನ್ನ ಯೂಸ್ ಮಾಡೋದ್ರಿಂದ ಏನಾಗುತ್ತೆ, ಎಲರ್ಜಿ ಆಗುತ್ತೆ. ಓಕೆ, ಅದನ್ನ ಇಲ್ಲಿ ಹೇಳಿದ್ದಾರೆ, ಇಟ್ ವಿಲ್ ಬಿ showing certain immune response in the human body. Now imagine if bacterium were available that could make proper human insulin, suddenly the whole process becomes so simple. So one can easily grow a large quantity of bacteria and make as much insulin as you need. Okay, ಇಲ್ಲಿ ಏನು ಗೊತ್ತಿರಬೇಕು ಅಂದ್ರೆ ಮುಂಚೆ ಎಲ್ಲ ಅದರ್ ಆನಿಮಲ್ಸ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಇನ್ಸುಲಿನ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ರು. ದೇ ವೇರ್ ಐಸೋಲೇಟಿಂಗ್ ಫ್ರಮ್ ಅದರ್ ಆನಿಮಲ್ಸ್, ಬಟ್ ಇಟ್ ವಾಸ್ ಕಾಸಿಂಗ್ infection, that is it was causing allergy. Okay, and would it not elicit an immune response in the question keltidare, which means it was eliciting the immune response, allergic reaction anna cause ಮಾಡ್ತಾ ಇತ್ತು. Now how to make this genetically engineered insulin? What was the challenge? How it was been done anta noduva. Think about whether insulin can be orally administered to diabetic patient or not. Why? ಸೊ ಓರಲಿ ನಾವು ಕನ್ಸ್ಯೂಮ್ ಮಾಡೋದ್ರಿಂದ ಇನ್ಸುಲಿನ್ ಅಥವಾ ಯಾವುದೇ ತರದ ಮೆಡಿಸಿನ್ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಇಟ್ ವಿಲ್ ಬಿ ಗೋಯಿಂಗ್ ಟು ಅವರ್ ಡೈಜೆಸ್ಟಿವ್ ಸಿಸ್ಟಮ್ ಅಂಡ್ ಇಟ್ ವಿಲ್ ಬಿ ಜಸ್ಟ್ ಪಾಸಿಂಗ್ ಥ್ರೂ ದ ಡೈಜೆಸ್ಟಿವ್ ಸಿಸ್ಟಮ್ ಆಸ್ ಇಟ್ ಇಸ್. ಸೊ ಯಾವತ್ತು ನಾವು ಮೆಡಿಸಿನ್ ಅನ್ನ ನಾವು ಎಫೆಕ್ಟಿವ್ ಆಗಿ ಯೂಸ್ ಮಾಡಬೇಕು ಅಂತಂದ್ರೆ ಇಟ್ ಇಸ್ ಬೆಟರ್ ಇಫ್ ವಿ ಟೇಕ್ ದ ಮೆಡಿಸಿನ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ಇಂಜೆಕ್ಷನ್, ದಟ್ ಇಸ್ ಥ್ರೂ ಇಂಟ್ರಾವೆನಸ್. ಓಕೆ, ಫಾರ್ ದ ಕ್ವಿಕ್ ಆಕ್ಷನ್ ಇಟ್ಸ್ ಬೆಟರ್ ಟು ಟೇಕ್ ಎನಿ ಮೆಡಿಸಿನ್ ಥ್ರೂ ಇಂಟ್ರಾವೆನಸ್ಲಿ. But however, some of the medicines, ಈಗ antibiotics ಅಂತದ್ದೆಲ್ಲ it has been taken orally. Okay, so insulin ಅನ್ನ ನಾವು ಯಾಕೆ orally ಅಷ್ಟೊಂದು administer ಮಾಡಿ ಕನ್ಸ್ಯೂಮ್ ಮಾಡೋದಿಲ್ಲ ಅಂದ್ರೆ because like it is more effective through the injection. Insulin used for diabetes was earlier extracted from the pancreas of the slaughtered cattle and pigs. Important to note. Insulin from these animal source could cause allergy. ಈಗಷ್ಟೇ ನಾವು ನೋಡಿದ್ವಿ ಅದರನ್ನ, and other type of reaction to the foreign protein. So insulin consists of two short polypeptide chain, that is chain A ಮತ್ತೆ chain B ಅಂತ ಎರಡು chain ಇರುತ್ತೆ ಇನ್ಸುಲಿನ್ ಅಲ್ಲಿ, and they are linked together by disulfide bridges. So insulin ನ ಸ್ಟ್ರಕ್ಚರ್ ನ ನೀವು ನೋಡೋದಾದ್ರೆ ಇಲ್ಲಿ they have got A peptide, B peptide, and there is going to be 2 sulfide bridges between A and B peptide. Very important, don't forget. Question ಬಂದಿದೆ ತುಂಬಾ ಸಲ NEET exam ಅಲ್ಲಿ. So sulfur ಏನು bridges ಇರುತ್ತೆ, 2 sulfur bridges ಇರುತ್ತೆ between A and B chain. Okay, so that is about the structure of insulin. So in mammals including humans, insulin is synthesized as a prohormone. ನ್ಯಾಚುರಲ್ ಆಗಿ ಪ್ರೋ ಹಾರ್ಮೋನ್ ಮುಖಾಂತರ, ಅಂದ್ರೆ ಪ್ರೋ ಎನ್ಸೈಮ್ ಹೇಗಿರುತ್ತೆ ಹಾಗೆ ಪ್ರೋ ಹಾರ್ಮೋನ್, inactive hormone which can be fully matured to produce active or functional hormone. So it is a pro hormone produce ಆಗುವಂತದ್ದು. Insulin is being synthesized as a pro hormone naturally in humans and even in other mammals. Okay, so which will be containing an extra stretch of protein, that is C peptide. So A, B, C ಮೂರು peptide ಇರುತ್ತೆ in pro hormone. So the C peptide is not present in the mature insulin and is removed during the maturation into the insulin. So main challenge ಏನಿತ್ತು recombinant DNA technology to make such insulin is that is assembling the matured form. Okay, so ಅದಕ್ಕೆ 1983 ಅಲ್ಲಿ ಎಲಿ ಲಿಲಿ ಅಂತ ಹೇಳೋ ಒಂದು ಕಂಪನಿ, ಅಮೆರಿಕನ್ ಕಂಪನಿ ಏನ್ ಮಾಡುತ್ತೆ, so they will be preparing two DNA sequences. They will be preparing 2 DNA sequences corresponding to A and B chain of human insulin, and they are being introduced into the plasmid of E. coli to produce insulin chain. So chains A and B were produced separately and extracted and combined by creating disulfide bonds to form human insulin. So A ಮತ್ತೆ B ಅನ್ನ separate ಆಗಿ synthesize ಮಾಡೋ ಹಾಗೆ ಮಾಡ್ತಾರೆ, and then they are being joined separately. So this is how human insulin was being made by Eli Lilly company, and this is very important. NEET 2021, NEET 2020 ಅಲ್ಲಿ ಕ್ವೆಶ್ಚನ್ ಬಂದಿದೆ. ಎಷ್ಟು ಸಲ್ಫೈಡ್ ಬ್ರಿಡ್ಜಸ್ ಇರುತ್ತೆ ಬಿಟ್ವೀನ್ A and B ಪೆಪ್ಟೈಡ್, ಮೆಚೂರ್ ಫಾರ್ಮ್ ಅಲ್ಲಿ ಯಾವುದೆಲ್ಲ ಪೆಪ್ಟೈಡ್ಸ್ ಇರುತ್ತೆ, and how Eli Lilly company has made the mature insulin, that is by separately making 2 DNA sequence to produce the peptide through the plasmid in E. coli. Okay, so this is what you have to know. Now the next concept, ಇನ್ನೊಂದು application under medicine, application of biotechnology under medicine is gene therapy. Gene therapy ಸಹ ತುಂಬಾನೇ important. NEET 2020, NEET 2022 ಅಲ್ಲಿ question ಕೇಳಿದ್ದಾರೆ from gene therapy. Okay, if a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy andre enu? It's a collection of method that will be allowing the correction of the gene defect that has been diagnosed in a child or embryo. So illi yen ಮಾಡ್ತಾರೆ, genes are being inserted into a person, cells or tissue to treat a disease. Okay, so correction of genetic defect involves delivery of a normal gene into an individual or embryo to take over the function or function and compensate for the non-functional gene. So gene therapy andre inserting the gene to make the defective gene to be normal. Okay, so that is gene therapy. So let's see the example. So ನೋಡಿ, 3 ಸಲ ಪ್ರತಿ ವರ್ಷ ಆಲ್ಮೋಸ್ಟ್ ಎವ್ರಿ ಇಯರ್ ನಾನು ತಗೊಂಡಂತಹ ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್, ಜಸ್ಟ್ ಪಾಸ್ಟ್ 5 ಇಯರ್ ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್, ಆ ಪಾಸ್ಟ್ 5 ಇಯರ್ ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್ ಅಲ್ಲಿ 3 ಸಲ ಜೀನ್ ಥೆರಪಿ ರಿಲೇಟೆಡ್ ಆಗಿ ಕ್ವೆಶ್ಚನ್ ಬಂದಿದೆ ನೋಡಿ. The first clinical gene therapy was being done in 1990 in 4-year-old girl with ADA deficiency. ADA full form adenosine deaminase. This enzyme is crucial for the immune system to function. The disorder is caused due to deletion of the gene for ADA. In some children, ADA deficiency can be cured. ಮುಂಚೆ ಎಲ್ಲ ADA deficiency ನ ಹೇಗೆ ಕ್ಯೂರ್ ಮಾಡ್ತಾ ಇದ್ರು? Through bone marrow transplantation and by enzyme replacement therapy. in which functional ADA is given to the patient by injection. So ಮುಂಚೆ ಎಲ್ಲ ಈ ತರ ADA ಅನ್ನ insert ಮಾಡ್ತಾ ಇದ್ರು into the patient. But the problem with both of these two approaches that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside of the body, and a functional ADA cDNA. ADA cDNA andre enu? cDNA andre enu? Complementary DNA. It is the DNA of the retrovirus, disarmed retrovirus. So a functional ADA gene anna ಈ ಡಿಸಾರ್ಮ್ಡ್ ವೆಕ್ಟರ್ ಒಳಗಡೆ ಹಾಕಿ ಅಂತಹ ಡಿಸಾರ್ಮ್ಡ್ ವೆಕ್ಟರ್ ಏನಿದೆ, ದಟ್ ಇಸ್ ಡಿಸಾರ್ಮ್ಡ್ ಪ್ಯಾಥೋಜನ್ ಇದೆ, ಮೇ ಬಿ ರೆಟ್ರೋವೈರಸ್, ದಟ್ ಇಸ್ ಬಿನ್ ಯೂಸ್ಡ್ ಆಸ್ ಅ ವೆಕ್ಟರ್. ಸೊ ಅಂತಹ ADA ಅನ್ನ ನಾವು ವಿ ಕ್ಯಾನ್ ಕಾಲ್ ಇಟ್ ಆಸ್ ADA cDNA. ಸೊ ಅದರನ್ನ ಇಟ್ ಇಸ್ ಬಿನ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಬ್ಯಾಕ್ ಇನ್ ಟು ದ lymphocytes. I repeat once again, lymphocytes from the blood of the patients are being grown in the culture. A functional ADA is being inserted into these lymphocytes, which are subsequently returned back to the patient. Adre problem enu, ee tarada lymphocytes enu produce madirtare, modify madirtare, so that is not mortal. They are not immortal, they are mortal, they will die, alva. So the patient requires periodic infusion of such genetically engineered lymphocytes. So that is the challenge with respect to gene therapy. Adre how gene therapy can be more effective? So gene therapy can be effective when the gene isolated from the marrow, that is bone marrow cells, producing ADA enzyme is introduced into the cells of the early embryonic stages. The permanent cure illa andre gene therapy is also not permanent cure. Okay, so this is what you have to know about gene therapy. Normally gene therapy can be treated with the two type of method: enzyme replacement therapy, bone marrow transplantation. And the next way is gene therapy. Gene therapy allu when it can be highly effective and permanent cure, when the gene from the bone marrow which is able to produce ADA enzyme, when that has been inserted into the early embryonic cells. So aga it is more effective. Illa andre periodic infusion of genetically modified lymphocytes is required. Okay, so illige we are done with another high weightage concept. Next innondu high weightage concept is molecular diagnosis. illinda 2023 NEET ಅಲ್ಲಿ ಕ್ವೆಶ್ಚನ್ ಕೇಳಿದ್ರು. 2021 ಅಲ್ಲಿ 3 ಸಪರೇಟ್ ಕ್ವೆಶ್ಚನ್ ಆಗಿ ಈ ಕಾನ್ಸೆಪ್ಟ್ ಇಂದ ಕ್ವೆಶ್ಚನ್ ಕೇಳಿದ್ದಾರೆ. ಸೊ ಮಾಲಿಕ್ಯುಲರ್ ಡಯಾಗ್ನೋಸಿಸ್ ಏನು? ಸೊ ಆಸ್ ಆಫ್ ನೌ ವಿ ಹ್ಯಾವ್ ಸೀನ್ ಅಡ್ವಾಂಟೇಜಸ್ of genetically modified plant, advantages of plants in the pest we have seen specifically, and we have seen how biotechnology has been used to make genetically engineered insulin. Matte medicinally en use, that is another use was gene therapy. And now let's see about biotechnology, use of biotechnology in molecular diagnosis. Okay, so ಯಾಕೆ ನಾವು ಡಯಾಗ್ನೋಸಿಸ್ಗೆ ಅಥವಾ ಟು ಡಿಟೆಕ್ಟ್ ಸರ್ಟನ್ ಡಿಸೀಸ್ ವಿ ಹ್ಯಾವ್ ಟು ಯೂಸ್ ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಂತ ಕೇಳಿದ್ರೆ, ಯು ನೋ ದ್ಯಾಟ್ for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. So using conventional method of diagnosis, andre serum, blood test, urine test ಎಲ್ಲ ಏನಿದೆ, so ಇದು early detection ಅನ್ನ ಮಾಡ್ಲಿಕ್ಕೆ help ಮಾಡೋದಿಲ್ಲ. We are not able to detect any disease in the very early stage through the urine or serum analysis. So recombinant DNA technology, polymerase chain reaction, ELISA. So idella kelavu techniques to serve the purpose of early diagnosis. Okay, so let's study all of this one by one, starting with maybe PCR. Presence of pathogen is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of the pathogen would go too much high, and it will be already causing lot of havoc in the body, and it will be causing the proper disease symptom. ಈ ಟೈಮಲ್ಲಿ ನಾವು ನೋಡಬಹುದು. ಬಟ್ ಸಿನ್ಸ್ ಆಲ್ರೆಡಿ ದ ಬಾಡಿ ಇಸ್ ಬಿನ್ ಡ್ಯಾಮೇಜ್ಡ್, ಕ್ಯೂರ್ ಇಸ್ ನಾಟ್ ಈಸಿ ಇನ್ ದ ಲೇಟ್ ಡಿಟೆಕ್ಷನ್. ಆದರೆ ಏನು, however, very low concentration of the bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplifying the nucleic acid by polymerase chain reaction PCR. So Very low concentration of bacteria viruses can be determined by amplification of the genes through the method called as PCR. Okay, ಯಾಕೆ ನಾವು PCR ಮೆಥಡಿಗೆ ಹೋಗಬೇಕು? Because in the case of certain viral diseases like HIV/AIDS, the late detection of HIV/AIDS won't be causing permanent cure. But if in case if we get to know about the HIV in very early stage, that is even when the symptom is not seen, so aga we can still try to treat the HIV/AIDS permanently, alva? So can you explain how PCR can be detected by very low amount of DNA? Idikin ee question ige answer previous chapter alli ide, alva? That is PCR reaction, PCR method anna ನಾವು ಕಲಿತಿದ್ದೇವೆ. 3 steps ಇರುವಂತದ್ದು. Extension will be using enzyme, first method will be using heat, so we already know about it. So PCR is now routinely used to detect HIV in suspected AIDS. ಅದೇ ತರ RT-PCR, which is a type of PCR, was being used to detect COVID, alva? So early detection of COVID. So COVID, if it was being detected early, it is also being cured effectively, alva? So it is being used to detect the mutation in the genes in the suspected cancer patients too. Yakandre cancer saha hage, in the early stage if we get to know that the patient is suffering from cancer, one can cure the cancer, alva? So it is a powerful technique to identify many other genetic disorders. So PCR has been used to detect HIV/AIDS. It is being used to detect any viral disease, by the way, and also the cancer. Very important. NEET 2021 ಅಲ್ಲಿ PCR related agi question ಬಂದಿದೆ. Next we have got about the next, that is autoradiography. Autoradiography is another molecular diagnosis technique. A single stranded DNA or RNA has been tagged with a radioactive molecule under a probe, and then it has been allowed to hybridize to its complementary DNA in a clone of a cell, followed by detection using autoradiography. So ಎಲ್ಲಾದರೂ ಇಂತಹ ಟ್ಯಾಗ್ಡ್ ಆಗಿರುವಂತಹ DNA ಅಥವಾ RNA, if they show complementarity with our DNA, it will be producing the light and we can say that our genes are normal. If in case if the photographic film doesn't produce any light, ಅಂದ್ರೆ ಏನರ್ಥ, ಈ ತರ ಟ್ಯಾಗ್ಡ್ ಆಗಿರುವಂತಹ DNA ಅಥವಾ RNA ಏನಿದೆ, that is not able to show complementarity with our DNA or RNA or gene, and there is the chance of cancer ಅಂತ ಅರ್ಥ. Okay, mutated anta, the gene that we have is mutated anta artha. So single stranded RNA athwa DNA use ಮಾಡ್ತಾರೆ, which is being tagged with a radioactive molecule. It will be hybridized with our DNA, so it will show complementarity if in case if our genes are normal. So the clone having the mutated gene will hence not appear on the photographic film, because the probe will not have the complementarity with the mutated gene. Okay, so this mutated gene can be anything, alva? So that is why they are not able to get show the complementarity. So ಏನು ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ ಏನು ಯೂಸ್ ಮಾಡಿರ್ತಾರೆ ಅವರು RNA ಅಥವಾ DNA which has been tagged with the radioactive molecule, so they will be having some common genes, so it should be showing the complementarity with the normal genes. Okay, so ಕೆಲವು genes ಎಲ್ಲ ಏನಿರುತ್ತೆ ನಮ್ಮಲ್ಲಿ, they are common to all of us. Okay, so if in case if we do not have that proper combination of genes, it means there is chance of our gene being different and there is chance of that one particular cancer anta. So that is how autoradiography is being done. Okay, next ELISA. NEET 2021 ನಲ್ಲಿ ELISA related question ಕೇಳಿದರೆ, this is another molecular diagnosis technique. Autoradiography is for detection of cancer. This is for cancer. Mutated gene andre it will be later causing cancer. ELISA, enzyme linked immunosorbent assay, is based upon the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigen like protein, glycoproteins as such, or by detecting the antibody synthesized against the pathogen. So this is being used to detect HIV. Okay, either the antigen will show the antibody, either the antigen that is produced by the pathogen will show the interaction with the antibody, or the antibody that we have produced against a particular pathogen will be showing the it will be like it will be reacting with the antibody or antigen that is present in the ELISA solution. Okay, so illige we are done with molecular diagnosis saha nodi aitu. And then we have transgenic animals advantage of transgenic animals enu anta. So transgenic animal, first of all, artha enu? Animals that have had their DNA manipulated to possess and express an extra gene are known as transgenic animal. So ಯಾವುದೆಲ್ಲ ಬೇರೆ ಬೇರೆ ತರದ transgenic animal ಅನ್ನ produce ಮಾಡಿದ್ದಾರೆ? Transgenic rats, rabbits, pigs, sheep, cows, fish have been produced. ಆದರೆ 95% of all existing transgenic animals are mice. Why are these animals being produced? How can man benefit? So that is what we will be seeing here. So there are five advantages we have got about transgenic animals. First advantage ಏನು? For normal physiology and development. Transgenic animals can be specifically designed to allow the study of how the genes are regulated, how they are affected, how they affect the normal function of the body and its development. Example, study of complex factors involved in the growth, such as insulin-like growth factor. So, idondu example. Bari example matra ನೆನಪಿಟ್ಕೊಂಡ್ರೆ ಸಾಕು related to the advantages of transgenic animal. So, ಒಂದು advantage ಏನು? To study the complex factors involved in the growth, such as insulin-like growth factor. By introducing such genes from other species that alter the formation of this factor and study the biological effect that result, information is obtained about the biological role of the factor in the body. Okay, so nothing more is important here. ಇಷ್ಟೇ ಸಾಕು, ಇಷ್ಟೇ ನೆನಪಿಟ್ಕೊಂಡ್ರೆ ಸಾಕು. ಈ ಪ್ಯಾರಾಗ್ರಾಫ್ ಇಂದ ಎಷ್ಟೊರೆಗೆ ಕ್ವೆಶ್ಚನ್ ಆ ಥರ ಏನು ಬರಲಿಲ್ಲ, ಬಂದರೆ ಇಲ್ಲಿಂದ ಬರುತ್ತೆ ಅಷ್ಟೇ. ನೆಕ್ಸ್ಟ್ is to study the diseases. So many transgenic animals are being used to increase our understanding about how a gene can cause a disease. These are specifically made to serve as models for human diseases, so that investigation of new treatment for diseases has been made. Today transgenic models exist for many human diseases. ಯಾವುದೆಲ್ಲ? ಕ್ಯಾನ್ಸರಿಗೆ ಇದೆ transgenic models to study these diseases like cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. So ಯಾವುದೆಲ್ಲ ಬೇರೆ ಬೇರೆ ಡಿಸೀಸಿಗೆ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಮಾಡೆಲ್ಸ್ ಇದೆ, that you should be knowing. ಬೇರೆ ಏನು ಅಗತ್ಯ ಇಲ್ಲ. The third advantage is biological product, that is to make biological product. Medicines required to treat certain human diseases can contain biological products, but such products are very expensive. So transgenic animals that produce useful biological products can be treated by introduction of the portion of DNA which will be encoding a particular product. So ಯಾವುದು ಪ್ರಾಡಕ್ಟ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ? ದಟ್ ಇಸ್ ಎಲ್ಲಿದೆ? human protein called as alpha-1 antitrypsin has been produced to treat emphysema. Very important. ಇದೇ ತರ ಬೇರೆ ಬೇರೆ ಏನು ಬಯಾಲಜಿಕಲ್ ಪ್ರಾಡಕ್ಟ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ ಟು ಟ್ರೀಟ್ ಫಿನೈಲ್ಕೆಟೋನ್ಯೂರಿಯಾ, cystic fibrosis. ಹಾಗೆ 1997 ಅಲ್ಲಿ ಫಸ್ಟ್ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಕೌ called as Rosie was being produced, which had human protein enriched milk, that is 2.4 grams per liter protein it had. The milk contained human alpha-lactalbumin and was nutritionally a more balanced product for human babies than the natural cow milk. So natural cow milk anna maguvige kodtare alva, adre ee milk alli it was having more nutritious compared to cow milk. Andre ee protein enide human alpha-lactalbumin, that protein is present in the human milk, so the baby had the very similar the baby was able to get the very similar protein through this Rosie milk, Rosie which is a transgenic cow milk. Okay, next vaccine safety ಹೇಳುವಂತದ್ದು fourth use of transgenic animal. So ಯಾವ ಏನು transgenic animal ಅನ್ನ vaccine safety vaccine ಅನ್ನ ಚೆಕ್ ಮಾಡ್ಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ? That is transgenic mice are being developed for use in testing the safety of vaccine before they are used in humans. So transgenic mice ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. ಸ್ಪೆಸಿಫಿಕ್ ಆಗಿ transgenic mice ಅನ್ನ ಪೋಲಿಯೋ ವ್ಯಾಕ್ಸಿನ್ ಅನ್ನ ಟೆಸ್ಟ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. So if successful and found to be reliable, they could be this transgenic mice has been replaced by monkeys to test the safety of the batches of the vaccine. Okay, ನಂತರ chemical safety ಅಥವಾ toxicity safety. So this is known as toxicity or safety testing. The procedure is same as that used for testing toxicity of the drugs. Transgenic animals are made that carry genes which can make them more sensitive to the toxic substances, so that they will, if in case ಒಂದು ತರದ animal, if it is more sensitive to a toxic, they will show quick function alva against that toxic, and we are able to study quicker. So adike transgenic models are being used, animals are being used to detect the safety of the toxic or chemicals. Aita, so yenide, transgenic animals are made that carry genes which make them more sensitive to the toxic substances than the non-transgenic animal. They are then exposed to the toxic substances and the effects were studied in toxicity testing, such as animals will allow us to obtain results in a less time. So that is what I just told now. So illige we are done with importance of transgenic animal. Nantra ethical issues ಬಗ್ಗೆ ಇದೆ, ಒಂದು ಸಲ ನೀವು ಓದಿದ್ರೆ ಸಾಕಾಗುತ್ತೆ related to ethical issues. So if you study by yourself also, it is enough. ಒಂದ್ಸಲ ನಾನು ಓದ್ತಾ ಹೋಗ್ತೇನೆ, ಏನಾದರೂ ಇಂಪಾರ್ಟೆಂಟ್ ಲೈನ್ ಇದ್ರೆ ಅಲ್ಲೇ ಟಿಕ್ ಮಾಡ್ತೇನೆ. ಆಕ್ಚುಲಿ ನೀಟ್ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಈ ಪ್ಯಾರಾಗ್ರಾಫ್ ಇಂದ ಕ್ವೆಶ್ಚನ್ ಬರಲಿಲ್ಲ ಇಷ್ಟವರೆಗೆ. The manipulation of living organism by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organism. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organism can have unpredictable results, such as organisms are introduced when such organisms are introduced into the ecosystem. Therefore, Indian government has set up an organization called as GAC. GAC a full form anna tilkondiri. GAC, Indian government ಅವರು set up ಮಾಡಿರುವಂತಹ organization which will make the decision regarding the validity of the genetic modified research and the safety of introducing genetically modified organisms for public services. The modification usage of living organisms for the public services has also created problems with the patents granted for the same. There is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic materials, plants and other biological resources that have long been identified, developed and used by the farmers and indigenous people. Andre biopiracy related ಆಗಿರುವಂತಹ ಕಾನ್ಸೆಪ್ಟ್ ಇದು. So this we have we will be seeing about this, however. So let's see about biopiracy. So biopiracy ಆಗಿರುವಂತದ್ದು like on rice, neem and turmeric. So rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. There are an estimated 2 lakh varieties of rice in India alone. The diversity of the rice in India, one of the richest in the world, basmati rice is a distinct for its unique aroma and flavor, and around 23 documented varieties of basmati rice are grown in India. There is reference to basmati in ancient text, folklore, poetry, as it has been grown for centuries. 1997 alli ಒಂದು American company, it got patent rights on basmati rice through the US patent. Through the US Patent and Trademark Office, that is UTO anta hagene barutte. This allowed the company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers variety. ಈ ಹೊಸ ವೆರೈಟಿ ಏನಿದೆ, ಏನು ಇವರು ಹೊಸ ವೆರೈಟಿ ಅಂತ ಕರೀತಾರೆ, an American company. So it is actually derived from the Indian farmers variety. Indian basmati was crossed with the semi-dwarf varieties and claimed as a new invention or a novel product. So it is an Indian basmati rice which was being crossed with a semi-dwarf variety and claimed as a new one. The patent extends to functional equivalence, implying that the other people selling, ಅಂದ್ರೆ ಬೇರೆಯವರು ಸೆಲ್ ಮಾಡೋದನ್ನ, ಬಾಸ್ಮತಿ ರೈಸ್ ಅನ್ನ ಸೆಲ್ ಮಾಡೋದನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡುತ್ತೆ ಈ ಪೇಟೆಂಟ್ ರೈಟ್ಸ್. ಸೊ several attempts have also been made to patent uses, products and processes based upon the Indian traditional herbal medicines, like ಇದೇ ತರ ಇದಕ್ಕೆ ಬಯೋಪೈರಸಿ ಅಂತ ಹೇಳುವಂಥದ್ದು. ಈ ತರ ನಮ್ಮ ಇಂಡಿಜಿನಿಯಸ್ ಪ್ರಾಡಕ್ಟ್ ಅನ್ನ ಬೇರೆ ಬೇರೆ ಕಂಟ್ರಿ ಅಥವಾ ಬೇರೆ ಕಂಪನಿ ಸೆಲ್ ಮಾಡೋದನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡುವಂಥದ್ದು, ಅಥವಾ ಇದು ನಮ್ದು, ಇದು ಹೊಸ ಪ್ರಾಡಕ್ಟ್ ಅಂತ, ನಮ್ಮ ಇಂಡಿಜಿನಿಯಸ್ ಪ್ರಾಡಕ್ಟ್ ಅನ್ನ ಹೇಳುವಂಥದ್ದಕ್ಕೆ ಬಯೋಪೈರಸಿ ಅಂತ ಹೇಳುವಂಥದ್ದು. ಸೊ ಈ ತರ ಬಯೋಪೈರಸಿ ಇಶ್ಯೂ ಟರ್ಮರಿಕ್ ಮೇಲೆ, ನೀಮ್ ಮೇಲೂ ಆಗಿದೆ. ಸೊ ಇಫ್ ವಿ ಆರ್ ನಾಟ್ ವಿಜಿಲೆಂಟ್ ಅಂಡ್ ವಿ ಡು ನಾಟ್ immediately counter these patent applications, other countries' individuals may encash on our rich legacy and we may not be able to do anything about it. So ಇದಕ್ಕೆ ಹೇಳುವಂಥದ್ದು ಬಯೋಪೈರಸಿ ಅಂತ. It is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and the people concerned, without compensatory payment. So that is about biopiracy. So most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. Most of the developed industrialized nation rich financially, ಆದರೆ ಅವರ biodiversity ಮತ್ತೆ traditional knowledge poor. In contrast, ನಮ್ಮಂತಹ developing countries ಏನಿದೆ, ಅಥವಾ developed or underdeveloped countries, they are rich in biodiversity and traditional knowledge related to the bioresources. So traditional knowledge related to the bioresources can be exploited to develop modern applications and can also be used to save time, effort and expenditure during their commercialization. There has been growing realization of the injustice and inadequate compensation and benefits sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation under biopiracy of their bioresources and traditional knowledge. So adike nama Indian Parliament yenide, so the Indian Parliament has recently cleared the second amendment of the Indian patent bills that take such issues into the consideration, and including patent terms, emergency provisions and research and development initiative. So this is about the chapter biotechnology and its application. ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ಯಾವ ಕಾನ್ಸೆಪ್ಟ್ ಇಂದ ಎಷ್ಟೆಷ್ಟು ಕ್ವೆಶ್ಚನ್ಸ್ ಯಾವ ಇಯರ್ ಬಂದಿದೆ ಅಂತ ತಿಳ್ಕೊಂಡಾಗಿದೆ. ಸೊ now it is time for you to study well and prepare well for your NEET 2024 exam, and hopefully you have liked the video. And that's all for today. Let's meet in the next class, next video. Until then, bye bye.